So, hello everyone, welcome and thank you for joining us on this episode of Spectroscopy in Action, our live webinar series. My name is Paula and I'm the Marketing Account Lead at Edinburgh Instruments. Today I will be joined by Iona, our application scientist, who will be presenting our webinar Accelerating Battery Design with Operando Raman Microscopy. Today, Iona will explore how Raman spectroelectrochemistry is driving innovation in next-generation battery design and electrocatalyst research. She'll introduce key building blocks, Raman microscopes, electrochemical cells, and potentiostasis, and show how they work together to deliver powerful real-time insights into complex electrochemical processes. At the end, we'll have a short Q&A section, so you'll have the chance to answer any questions you might have. Hope you enjoy this webinar, and I'll now pass over to Iona to get started.
Okay, sorry about that, a few technical issues, but today we're going to be discussing accelerating battery design with Operando Raman Microscopy. In this webinar, we're going to cover why you'd use Raman microscopy for battery analysis, how you might want to configure your Raman microscope for these measurements, and then we're going to go over 3 different case studies. The first looking at an operando measurement, the second looking at electrode degradation, and the third looking at the composition of an electrolyte. So firstly, why would you want to use Raman microscopy for studying electrochemistry? Raman is really versatile in that you can analyze a wide variety of material types. This can include metal oxides, carbon, aqueous electrolytes, and organic electrolytes and gels, all components of a typical battery. In that case, you can analyze the electrode, the electrolyte, and look at ion transport. When looking at the electrode, you might be interested in formation of new phases, structural changes, and degradation products. For electrolytes, you might be interested in ion solvation, interfacial analysis and hydration levels. And finally, you can look at insertion and extraction of ions, strain on electrode materials, and reversibility. So there's 2 kind of key components from the electrochemical side, and that is the electrochemical cell and the potentiostat. The cell is quite commonly a 3-electrode cell where you're testing the working electrode under electrochemical conditions. This also has a reference electrode, which has a stable potential reference, and a counter electrode, which balances the current going to the working electrode. The next is the potentiostat. This drives the electrochemical reaction, but must be compatible with the cell. It sets the desired potential between the working electrode and the reference electrode, and if deviations occur, it adjusts the current between the counter electrode and the working electrode to maintain the potential difference. So moving on to Raman spectroscopy-based electrochemistry, we want to couple these 2 components with our Raman microscope. That will allow us to apply a potential across a sample while simultaneously collecting spectroscopic data. In order to perform measurements, we'd have our material of interest in our working cell. This would be placed on the microscope, and the laser would be focused on the sample through the window of the cell. This allows real-time vibrational response of the material to an applied electrical stimulus, and we're able to track electrochemical transformations in our energy system. It provides time and spatially resolved analysis, and it allows us to understand the mechanics behind the electrochemical observations we're seeing. So a Raman spectrum gives you a wide range of information, and it's unique to the kind of sample type you're analyzing. It commonly has narrow bands, which can tell you about chemical structure. You can look at new phases, the introduction of impurities and degradation products. The intensity of your Raman is connected to your concentration, so you can monitor the concentration of your material based on intensity variations. The band position variation can tell you about strain in your sample. And finally, the band width variation can tell you things like crystallinity, dopants, and solvation structure. The Raman spectroscopic measurements can also be performed as an image. This is where a Raman spectra is collected at each pixel of a predefined grid across your sample in order to give you spatial Raman information about changes across your sample. This can be performed both in situ, so inside of the electrochemical cell, or ex situ after the material is removed. So that's a little background of why you might use Raman, but there's some key considerations when setting up your Raman microscope for these measurements. These are the basic components of your Raman microscope. First, the laser is used as your excitation source. This is directed through the microscope with a beam splitter down your objective lens onto your sample. So in this case, it might be your electrochemical cell on your microscope stage. The scattered light then travels back up through the microscope through a Rayleigh rejection filter. This filters out any elastic scattering and only leaves your Raman scattering of interest. In our systems, they're both truly confocal. So this then travels through a confocal pinhole, which the width can be adjusted to control the spatial resolution. The scattered light then enters the spectrograph, where it hits a diffraction grating. This disperses the light into different wavelengths, and these hit the CCD camera detector. And this is what changes your photons into an electrical signal, so you get your Raman spectrum. Although there's a lot of components, there's a few kind of important components which will influence your spectral and imaging outputs of your experiment. These are the laser, the grating, and the detector. So let's discuss these in a little more detail. So when selecting your laser, this is really going to be down to sample specific, but you want to find a balance between the signal intensity, fluorescence, spectral resolution, and spatial resolution. First of all, the intensity is proportional to the wavelength. So the shorter laser wavelengths, you would have a higher intensity. Another key factor is the fluorescence. So you can see in this spectrum here, the spectra collected at 532 has a large fluorescent background, which overall masks some of the Raman peaks. When moving to the 785, this fluorescent isn't seen. So in general, when you want to avoid fluorescence, you'd move towards a near-infrared laser because fluorescence is normally seen within the visible region. Finally, the spectral and spatial resolution will also affect the wavelength that you select, with higher spatial resolution as you move to UV and higher spectral resolution as you move to NIR. There's one other kind of key factor when looking at lasers, and that is resonance. You want to choose a wavelength that's near an electronic transition of your material to give you resonance Raman. This will dramatically enhance the signal, while off resonance in comparison will give you weaker scattering. So an example of this is choosing a 532 laser for the sp2 transition in carbon, as that is close to the electronic transition. So moving on to the gratings, this can control the spectral resolution. So the lower density grating, so the 300 groove, for example, you can see that the spectral resolution is low, whereas moving up to the 900 and the 1800, you're getting higher spectral resolution. This is a trade-off. When you move to your higher groove density, you will also collect a lower spectral range. So these two factors are really important to consider when picking your grating. The final choice is the detector choice. Normally in Raman, a CCD camera is used. A standard front illuminated CCD, as you can see here, peaks at around 55% quantum efficiency, whereas a back illuminated option increases that quantum efficiency up to about 95%. The reason for this is the front illuminated CCD, the photons must travel through the electrode and insulating layer before they reach the photon sensitive area, and this leads to light attenuation. On the other hand, the back illuminated, the photons shine directly onto the photosensitive silicon, which is why you get your higher quantum efficiency. This becomes very important when you're thinking of mapping your sample. So here we have the same sample analyzed with 532, 785, and 830 nanometer lasers, both with the back and the front illuminated CCD. And you can see that in all cases, the back illuminated has much higher signal intensity, with almost 3.2 times higher in the 532, about 4.9 times higher in the 785, and about 4.3 times higher in the 830. As I mentioned, this is really important consideration for mapping. So the maps on the right-hand side, the top one is your front illuminated CCD. It required a spectral acquisition of 0.5 seconds, which led to a total imaging time of around 2 hours, or just under 2 hours. Using a back illuminated CCD, the acquisition time could be reduced to 0.2 seconds to give a similar relative intensity, which led to the mapping time being reduced to almost 50 minutes, so almost half as much. So that shows how the back illuminated CCT gives high sensitivity, which leads to faster imaging. When thinking about faster imaging, another option is your EM-CCD. This uses multiplication gain technology, which increases the number of electrons generated by a single photon in order to increase your signal. As you can see from the spectrum here, the gain can be controlled. So with zero gain, it acts as a kind of standard CCD, and the signal is quite low. And as you increase up to 200 gain, the signal intensity increases up to around 50 times. Now moving on to our systems at Edinburgh Instruments, we have the RM5 and the RMS1000, which are both confocal Raman microscopes. Firstly, the RM5 is our kind of smaller benchtop research grade instrument, which can perform both Raman and spectral PL. This can have up to 3 internal Raman lasers, which can cover the visible to the near infrared range. On the other hand, our RMS1000 is our larger multimodal confocal microscope. As well as Raman and spectral PL, it can also be used for other imaging techniques such as single harmonic generation, 2-photon excitation, and FLIM mapping as well. For it to do this, it can couple up to 5 internal lasers, which can be used for Raman, but can also couple external lasers such as pulse lasers needed for some of these imaging techniques. Although our RMS1000 is very kind of multimodal and customizable, for these type of studies in electrochemistry, the RM5 would be a really good choice of microscope to go for. Firstly, as I mentioned, it can hold up to 3 lasers, going from 405 to 1064. It has an encoded stage allowing for mapping in X, Y, and Z, and this can be coupled with either a custom or a home-built electrochemical cell. Both of our systems are truly confocal, so have an adjustable pinhole to control spatial resolution. It can hold up to 5 diffraction gratings, giving you that control over spectral resolution, and that will cover the range of lasers that you might have in the system. And finally, it can hold up to 2 detectors. So the first might be a CCD, for example, the back illuminated CCD for high quantum efficiency. And secondly, you might want to couple in an EM-CCD for fast mapping. So that's the kind of background covered. I then have 3 case studies to go over. All of these case studies are either customers that have carried out their research using our instruments or collaborations we have with customers where we've also carried out the research. This first example is Operando Raman Microscopy of water splitting electrocatalyst. In this case, the customer was measuring an electrocatalyst that they developed, which is nickel iron phosphide, and they wanted to check its efficiency versus alternatives. For this, they used the RMS1000 coupled with a custom 3-electrode electrochemistry cell. To give a little bit of context, they were interested in an anion exchange membrane water electrolysis system. This is a green hydrogen for green hydrogen production, which splits water using renewable electricity with 0 CO2 emission. The membrane conducts OH ions and separates the hydrogen evolution reaction with the oxygen evolution reaction for efficient and safe operation. It allows for a compact design, which uses low-cost transition metal catalysts, although optimization of this type of system would be crucial for commercial scale-up. That's where Operando Raman comes in, which can offer mechanistic insights into the catalyst surface and the electrochemical performance. So first, they synthesized and characterized their material. The synthesis, they did hydrothermal growth of nickel iron layered double hydroxide on a nickel foam. This then went under gas phase phosphidation to create the nickel iron phosphide. They wanted to characterize this material, so they first used the RMS1000 with a 532 laser and an 1800 groove density grating for high sensitivity and high spectral resolution spectrum, which you can see here. The Raman confirmed iron oxygen and nickel oxygen vibrations, which suggests oxidation. And it also showed the metaphosphate ion group at 680, which is the evidence of the phosphorus inclusions in the material. So they could use this to assess the successfulness of their synthesis. Once they were happy with that, they could move on to Operando Raman. Once again, they used the RMS 1000. This was coupled with a custom 3 electrode cell. They collected spectra at different potentials with a 200-second acquisition. Their home-built 3 electrode cell had a working electrode of either the nickel ion LDH or their kind of newer catalyst, the nickel iron phosphide, because they wanted to compare these two. The counter electron was platinum. The reference electrode was silver chloride, and they scanned the potential up to 1.7 volts in order to see how their system operated. What they were seeing from the electrochemical chemistry data is that the nickel iron phosphide had superior hydrogen evolution reaction performance compared to the nickel iron LDH, but both catalysts had a similar OER performance. So they wanted to understand why that was the case. So first, they performed the operando measurements with the nickel ion LDH. What they saw was that Ni2 plus oxygen diminished. So this is the orange bands here. And the Ni3 plus became more intense, which is shown in green. This was evidence of oxidation of the catalyst, which might explain the kind of performance of the oxygen evolution reaction. They then went on to their kind of more novel catalyst for analysis. They carried out the same analysis, and what they saw that was that up to 1.4 volts, the Raman spectra recorded were very similar to the ones I showed in the last slide. So the characterization remained unchanged. However, after 1.5, the bands corresponding to the metaphosphate anion dampened, along with the Ni2 plus also diminishing. The Ni3 plus became more prominent, and these were all indication that after this voltage, the material became unstable. So overall, this study provided evidence that the phosphides are unstable under electrochemical oxygen evolution reaction, and explained why they didn't see a heightened performance in their new catalyst. So overall, the Raman could be used to understand how these materials operated. So overall, for that study, they used the Raman to enable surface-specific operando analysis to track real-time chemical changes in their electrochemical cell. They used the RMS 1000 coupled with a custom 3-electrode cell. And this type of experiment is ideal for understanding the mechanisms required for material discovery and battery design. A link to that case study, I just wanted to show a quick software feature. Ramical is our Raman software for both of our instruments, and I wanted to show this external trigger mode feature. So this is where instead of an internal trigger from, for example, you telling the software to start spectral acquisition, it's taking an external pulse from an external hardware, such as a potentiostat, to control the start of the analysis. So I just have a little video here. This is the kind of basic setup of your measurement parameters, where you can select your laser power, your gratings, things like your pinhole and detectors. So once that's set up, you then go down to your measurement window, and you can see that internal is selected. This means that when you go to start, your measurement is instantaneously collected. This is the same when you go to any of the other kind of imaging modes, such as live. You start the live, and that triggers the measurement to be collected. And there's no delay between clicking start and that measurement. That's the same in kinetic mode as well. What you'll see in a second is switching to the external trigger mode. And as I said, this is using a pulse from the external hardware to trigger the spectral acquisition. So now when clicking live, there's a delay before the measurement is acquired, and that's because the software is waiting for that external trigger before it starts measurement. This is the same when just taking a single measurement. the start button will be selected, and then there's that delay waiting for that pulse. So this is really useful when setting up, for example, an operando measurement, because you could do a kinetic scan where you have an external trigger pulse from your potentiostat, and you set your measurements to be at certain points. So really good for kind of a batch analysis for operando measurements. So now that that's started, this has been set to collect 20 different measurements. And like I said, this is taking a pulse from the external source. If I just skip forward until the measurements have been collected, what you can then do is visualize all of these measurements, and then you can correlate them with the hardware trigger profile. So in that case, you could see how your potential is changing your Raman signal, for example. So this is a nice way to incorporate those operando measurements through our software. So moving on to case study 2, this is imaging structural changes in an electrode after cycling. In this work, our customer was looking at a photo-rechargeable battery. These are have dual functionality because they harvest solar energy as well as storing it directly as chemical energy. This dual functionality is achieved through the photoelectrode because it performs 2 roles. The first is to act as a light harvesting material, and the second is to simultaneously function as a standard electrode within the battery. In doing so, this combination imposes quite a high performance demand, which requires like a long cycle life as well as the battery being very robust, because it must withstand both simultaneously electrochemical cycling as well as photoexcitation from light harvesting, and that introduces a very unique and accelerated degradation pathway. So here they want to use Raman to understand the mechanics behind this and check the degradation and deposition of the electrode. For this analysis, an RM5 microscope was used with a 532 laser, a 1200 groove grating, and a CCD camera. The electrodes were imaged in 2D, and the non-negatively squared method was used to look at the components within the maps. To give you a bit of background about the electrode material, these are titanium dioxide reduced graphene oxide electrodes. These contain different phases of the titanium dioxide. And what they were interested in is first a pristine uncycled electrode, and then comparing it to an ex situ cycled electrode. So this was cycled with 3 volts for 500 cycles. And then these images were compared. So first of all, here is a map of the pristine electrode. This provides a baseline for the spatial distribution of components in the electrode. There was 3 main components that were found. Firstly, rutile, which is in green here, anatase in red. So these are both phases of the titanium dioxide. And then finally, reduced graphene oxide, which is in blue here. In this case, the conventional Raman maps that were collected did show broad spatial distributions, but they suffered from overlap between the titanium dioxide and reduced graphene oxide components, which is why this analysis was performed with non-negatively squared. This deconvolutes the overlapping signal for clearer component visualization across the electrode. So as you can see, the maps show that the pristine electrode had a dominant anatase, which was uniformly distributed across the surface. So that's shown in red here. There was also the rutile phase, but this was more clustered and less uniform across the electrode. And finally, the reduced graphene oxide formed fibrous conductive networks across the electrode as well. Finally, at the bottom here is the non-negatively squared residuals map. This is used to validate the chemometric model. And as you can see, a low intensity across the entire field confirms a good quality of linear fit in this case. So now that they had a baseline for their pristine electrode, they then wanted to compare it with the cycled electrode, with the goal to assess any compositional changes after extended operation of the electrode. As I mentioned, this electrode was cycled 500 times, and then a Raman map was collected in the exact same way as the pristine electrode. The maps here are presented as a 3-component and non-negatively squared overlay map in the same colors as you previously saw. Once again, the pristine electrode showed a uniform anatase region with clustering of the rutile and fibrous networks of the graphene oxide. You can see quite a drastic change in the cycled electrode. This time, there is a pronounced loss of that green rutile signal, and the cycled electrode is dominated by the anatase and the reduced graphene oxide signal. This is evidence that the rutile is selectively degraded, and this also tells us that the rutile is less structurally and electrochemically stable than the anatase phase of titanium dioxide after repeated cycling. We can also see in the bottom image, the cycled electrode, a kind of blue circle at the top. This shows our cell seal edge, so where the electrolyte was present and where it wasn't. And we can see that above this line where the electrolyte wasn't in contact with the electrode, we still see our rutile green signal. So this confirms that the degradation of the rutile was directly linked to the electrochemical cycling rather than just intrinsic instability of the material. We're also interested in studying the any deposits on the electrode after cycling. So this battery is a lithium hexafluorophosphate electrolyte used in the battery. So it was anticipated that the deposits would be electrolyte salt on the surface. When looking at the spectra from the cycled electrode, we also saw a band at 748, which is a band assigned to the hexafluorophosphate anion. This was mapped across the area, and you can see that this salt appeared in localized kind of positions across the electrode, which confirms solid precipitation did occur during cycling. It can also be seen, so this is the exact same electrode area as I showed on the previous slide. You don't see the difference between the cell sealed area and outside of that. So it does suggest this was seen kind of all over the electrode, which is explained by post-cycling migration, and it actually highlights a key challenge with ex situ analysis like this for capturing the true distribution of some of these components on the electrode. So this maybe highlights why an operando style measurement would be superior. So overall, the Raman, along with the chemometric analysis, was a powerful tool for investigating the electrode structure. It also highlighted the loss of electrode phase, as well as the electrolyte salt precipitation. This shows that Raman can be really useful to show degradation of both the electrode and the electrolyte pathways. And carrying out this can then inform the design of a more durable and efficient photoelectrode. I next wanted to show you another software highlight that was linked to this case study. This is our Python IDE. This connects Python through our own software so that you can either control the electronic hardware using the Python, or you can perform data processing using this Python as well. So if I just go on to this video here, you'll see that this is the electrode map that I showed you. It's just showing an intensity map across the whole spectrum, so it's not really telling us any useful information at this point. And as I click around, you can see the different spectra at different points of the map. I can then go to the Python IDE at the top here, and this pulls up our Python screen. So here you can load any Python scripts you like, which can, like I say, do processing of the data, and it can be ran straight here. What you can also do is, instead of going to the Python IDE, you go to the favorites menu, which is next to it, and here you can have all your favorite scripts loaded, ready to go. So for this one, I've run the non-negatively squared chemometric analysis, and what it's doing is it's sending the ramical data in the background to Python, where it's running this analysis, and then the results will be pulled through into the ramical software. So if I just skip ahead until that's performed, what you'll then see is the fraction maps, as I showed you in the previous slide. They're now appearing through in ramical. So that means you can then kind of save this with the raw data, as well as changing the visualization, such as color schemes and things like that. So you have more control over the kind of data processing and manipulation of your data. Okay, so that brings me on to the third and final case study. So we've looked at operando measurements as well as imaging of the electrode. Now this study is looking at the composition of an electrolyte, and in particular, how that electrolyte can be changed in order to inhibit ice formation. So this study is based on flexible zinc air batteries. These are low-cost, safe, environmentally friendly batteries, which have a large theoretical energy density. And they're of particular interest for applications such as wearable technologies, medical devices, and aerospace components. However, one of the main challenges with these types of batteries is that they have their mechanical properties and conductivity decrease at low temperatures, which reduces the efficiency of these batteries at sub-zero temperatures. These researchers were looking at a solution to that by incorporating lithium chloride into the cellulose nanofiber-based hydrogel electrolyte. As they expected, this would inhibit the ice crystal formation, which reduces the efficiency of the batteries. Just a bit of background about the kind of specific battery. They looked at a sandwich-structured flexible zinc air battery, which had a iron cobalt nanosheet catalyst as the air electrode and a zinc anode. As I mentioned, they were using this novel CNF-based flexible hydrogel electrolyte, and this incorporated sodium polyacrylate as a cross-linking network, and also lithium chloride, which they were hoping to use to inhibit ice crystal growth. They then used Raman in order to characterize their hydrogel, but also to have an idea of the performance of the hydrogel as well. So for the Raman analysis, they used an RM5 equipped with a 532 laser. They studied a very wide spectral range from 400 to 4,000 wave numbers. The reason they did this was to look at the fingerprint region of the electrolyte in order to characterize the material, but they were also interested in water vibration modes, which appear kind of 3,500 wave numbers. And that was in order to get an idea of the performance of the electrolyte. So looking at the characterization of the material, what they saw was with increasing lithium chloride concentration, they also saw an increase in intensity of CH bending modes. They saw a decrease in intensity of CH2 twisting and C double bond O stretching modes. And finally, they saw a splitting of the CH2 bending mode from one peak into two. All of these changes were attributed to the chloride ion embedding in the hydrogel structure and thereby altering the hydrogen bonding. So using Raman, they were able to confirm the morphology of their electrolyte, but also see the effect of how varying the lithium chloride concentration had on the spectrum as well. They then wanted to understand the process that was occurring when this lithium chloride was introduced. And to do that, they wanted to look at the water vibrational modes, in particular two key OH stretching bands. The first, the symmetric stretch, which was at 3263. This is dominant in rigid structures like ice. The second was the asymmetric stretch at 3433, which is dominant in water or more like flexible structures due to forming and breaking of hydrogen bonds. To assess this, they looked at the ratio between these two, and they saw that with increasing lithium chloride concentration, they saw a reduced intensity ratio between the symmetric and asymmetric. And what this meant was that the asymmetric was becoming more dominant with increasing lithium chloride. This overall showed that the lithium chloride weakens the water's ability to form ice crystals and thus inhibits the ice formation at low temperatures in these batteries. So just to summarize, they were able to use Raman to characterize the hydrogel structure, as well as how this inhibits the ice forming mechanism. They saw that the lithium chloride prevented this ice formation, which is crucial for these flexible zinc air batteries to be used at low temperatures. And understanding the mechanism behind this, which Raman was able to do, enables them to develop more flexible batteries for aerospace components and things like wearable technologies that are to be used at sub-zero temperature. So that really brings me to the end of the case studies there. As I mentioned, we went over a operando measurement using Raman. We looked at the degradation of electrodes, and finally the composition and effect of electrolyte changes in a battery. Overall, the RM5 would be a really nice microscope to use, which can be coupled with electrochemical cell and potentiostat for operando Raman measurements. The versatility of Raman can make it very good for battery design by studying electrodes, electrolytes, and ion transport. And overall, incorporating the Operando Raman microscopy can aid with battery design and really understanding the mechanisms behind the electrochemical performance. So thank you very much for your attention, and I'll take any questions.
Yeah, got it. Yeah, sorry about that. More technical difficulties today. It's really not been a good day for us, has it? So yeah, thank you, Iona. That was really interesting. As I said, I did say the question box is now open if anyone has any questions. So we have one that had already come in. So that was, can you track the formation of solid electrolyte interface in situ with Raman?
Yeah, absolutely. So the solid electrolyte interface could be monitored using Raman. For example, in a lithium battery, you're going to get species like lithium carbonate and lithium fluoride and things like that at the interface, and that could definitely be distinguished by Raman because each of those kind of species have a very unique vibrational mode. They'll also appear kind of after cycling, so you could maybe track those using the operando measurements, like I mentioned. So definitely.
Thank you. The second question that we have is, can Raman help us diagnose a specific failure, like a battery that's losing capacity quickly? And what would the Raman fingerprint of a failed electrode look like?
Yeah, so I think the case studies I showed had some nice examples of how you could look at battery kind of failure. But I think one thing to consider is it could be multiple factors that are causing the battery to fail. So I think being able to use Raman to look at the electrode as well as the interface and the electrolyte can really give you a better kind of idea of all components that could be causing a failure in that battery.
Okay, thank you. So, unfortunately, because of the issues we had earlier, we don't have a time for any more questions. But if there is anything else you would like to ask, you can email us at inquiries@edins.com for any further questions that you have. So, thank you to Iona for telling us about Raman microscopy, accelerating battery design today. This webinar will be published on YouTube for you to watch again and share with colleagues if you think it will be useful for them. And we will be continuing our series next month. It'll be our final webinar of this year. So make sure you're subscribed to our newsletter to be the first to find out about that. And also, if you are subscribed to our YouTube channel, you'll get notifications about all of our latest videos. So finally, thank you to everyone for joining us. We hope you enjoyed this session and found it useful, and we will see you next time.